Welcome to the third in this series of our Neuroimaging Research Group fMRI workshop. And in the last couple sessions, we covered a lot of material about the structure of the imaging files coming from the scanner after we run tasks, the accessing and processing data from presentation software like ePrime. And then we started in on some of the initial processing steps for our actual neuroimaging software suite, which we're using AFNI in this example. There's a lot of general principles that I've been presenting on that are applicable to any neuroimaging software, but that's the example we specifically use here. So there's two things that I definitely want to cover within this session, and then if we have a little bit of additional time, I'll start going into quality control review. So one of the really nice things about AFNI is it has a couple of these Python scripts. So .py, just as reference, is generally the extension that's used for anything that's programmed in Python. It's not required in a Linux environment, but that's standard practice. So this AFNI_proc stands for processing, and that's a program that ends up helping to build your whole processing pipeline. And it's nice because it's automated and can be applied very precisely in the same way to all of the data that you're interested in running. So we'll spend a long time on that. Most of this program has to do with processing raw data into a format that's usable and analyzable with other subjects' data. So most of this program, what it's doing is all of the pre-processing steps. And then at the very end of the program, it tags on the level 1 regression. Regression meaning in this context that we're using information from our ePrime behavioral data to look at brain activation during particular parts of the task. Level 1 here ends up referring to the level of individual subjects, to be contrasted with level 2, which is looking at either groups, it's looking at groups of people, either in relation to other groups or in relation to variables of interest. And so that's when we talk about level 2, or if you hear me say level 2, that's what I'm referring to, whereas level 1 is just one person. And what's nice is there's also some tools output as part of this Python program that sets us up to do some basic quality control. It doesn't quite cover everything that you would probably want to do, and there's probably some specifics to each experiment, but it's a great place to start, and it has you actually looking at your data, which is, as I belabored a lot in the past two sessions, it's really important to look at what you're actually putting into your program, because there's a lot of really complex things, or the data structures themselves are very complex, and if you put them through a lot of this pre-processing, the data will look great even if it's actually not. So it's good to look at all these steps. So I definitely want to spend time on that. We'll see if we get to that in this session, but if not, session 4 will cover a lot of that. Okay. One thing, there's a couple of things that I think that are really helpful within the paradigm that we're using. And while AFNI as a program can get a little complicated sometimes, their documentation system is really good. So within this website, and we can save this, but I'll. Okay. If we look at this, so this is the AFNI website. It's afni.namh.nih.gov. And this is their program helps page, which you can access always here from the main site. This is a list of all of the programs that are part of AFNI. And this is complex programs as well as little modules that are really used in other programs for very specific goals. But within each of these, there's a, if you click in here, there's usage directions about how you use it. It tells you what it does, and it gives you a list of all of these options that you can do when you're running the program. Now I've just picked one at random, but you can see even for this one program, there's probably 20 pages or so. This is a pretty good resource. It can take a while to find something here, but pretty much all of the documentation tends to be present. So if I jump down here to AFNI ProcPy, so AFNI ProcPy is a big program, so much so that it has a bunch of different tables of contents, including examples and really extensive notes sections. And you'll note if I scroll here, the scroll bar has gone about this far. This is, maybe 150 pages or something long. So when you're reviewing these, it's really useful if you're looking for something specific to use the find functions in here. So you can find something like if we're interested in TCAT, for example, it can quickly jump me to where I need to go. So I don't want to spend too much time on here, but this can allow you to look into, I'm going to do an overview of each of the steps, but if you want more detail or if it's something that you're interested in changing or altering, this is a really good resource to use. And a little later, there's another link in the presentation to the program specifically. Talking a little more broadly about AFNI ProcPy. So, like I was saying before, this is a program or a script written in Python, and essentially what it does is it bundles all of the common commands used in pre-processing into a series of steps. And so it's really kind of a wrapper program that generates a script in T shell, which is the C shell in Linux that's also commonly used. And basically what it does is it's a program to generate these T shell scripts that will actually run your data for a single subject. And there's a couple of things that are nice about that. One is if you need to make small alterations or changes for a given subject, you don't need to hand edit a really long script. You can just change some options within AFNI ProcPy and it'll generate the right script that you want. The other thing about this, though, is that it's pretty flexible. So if you use AFNI ProcPy to generate the script for your level 1 analysis, but you want to hand edit it, you can use a copy of the script that it generates, which is called proc dot whatever subject number you have. You can use that and then edit it manually if you wanted to add special steps or little special subscripts into your processing pipeline. I like to use AFNI ProcPy primarily on its own to generate the script that I want every time on the fly when I need to run a new subject. I find that it works better for standardizing the analysis. And so, yeah, here's the link to the extensive documentation for AFNI ProcPy. included in there are also examples for particular types of analyses. So let me go ahead and step out a little bit. So just as a quick review, there's three main programs that we tend to use when working on the cluster. There's this program called XMing, and what that does is it allows this other program, PuTTY, here, which is a kind of just a command prompt. If you run a program on here that uses a graphical user interface, that is a window that you can interact with, XMing allows data from our supercomputing cluster to go back to this computer so that it can make a screen that you can interact with. PuTTY is a program, and if you want details, you can refer back to one of the earlier sessions, but PuTTY is a program that lets us log into the computing cluster from an NCH computer and run programs and commands remotely on another computer, essentially. So I have set up here right now this this PuTTY window. So this is what I would use. This command prompt is what I would use to run AFNI-ProcPy. For our purposes, though, I find it can be a little bit harder to read the AFNI-ProcPy commands in this window, so I'm using another program. This is called WinSCP. This is just an FTP client that allows us to look both here locally on my desktop at my data and over here remotely on the computing cluster. So last time we went through a little bit of the pre-processing of our raw data that was here, and that ended up yielding these head and brick files, which contain both anatomical images and epi images of 4 runs of a task that we were interested in. And that ended up going in here. This is where we end up putting our subject data. It's in a separate folder organized by task. The subjects and our subject number, in this case I've just called it demo sub. In this AFNI folder is where we tend to put our raw images. So to move that over, I can just go into our raw data, select all of these, and use the duplicate command. Okay, so if I go to go, no go, subjects, no subject. And this command just lets me move my data files over. You could copy them any other way you wanted to. Okay. And this is just, you can use the FTP client to move things. You can also use this command prompt over here. I tend to use WinSCP for when I'm doing more complicated file moving and when I really need to look at like a lot of information about when files were changed and what permissions are available, so like who can access the file. So for this step I tend to use WinSCP. And basically those image files, in that same directory there's also output from our scripts that work on behavioral data in another folder there called onsets, which contains timing information about what happened during the task. So these are the two main components and pretty much all you need in order to run a subject in this framework. So if we look in our subject folder here, AFNI here now contains our head and brick files. These are the these are the images from the task itself in a format that AFNI can read. And then in here too, we have an onsets folder, and just a rapid review from this time. This file is a simple text file, but it contains the timing information of when a particular event occurred during the task. And I have one of these files for each of these conditions. In our case, the task that we're using is the go, no-go task, which contains 4 conditions based on whether they responded correctly to either a cheese being presented so you should push the button, that's correct go. If you don't push the button when the cheese shows up, that's an incorrect go. I may have, yeah, anyway. But each of these summarize, if you click during the cat, which you're not supposed to click, that goes into one file, and then if you correctly don't click when the cat shows up, that's in a file too. Sorry, that gets a little bit jumbled, a lot of false or double negatives there.
Yeah, so just for a little bit more cloud level, with fMRI, the signal to noise is pretty small. So what you have to do is have lots and lots and lots of events in order to sort of filter out what is noise from what is signal. So with fMRI studies, you tend to have, you try to design a task that has lots and lots of different psychological events that are happening, and then see what's happening on average in the brain across all those. So for this study, we're having it's a real simple study where we have a go event and a don't go event. So that's basically what those are.
So our imaging files are located here in this AFNI folder within our subjects demo subject folder. This onsets folder has our behavioral data, and this right now is an empty folder called results. And that's a folder that I made, and that's where our output will end up going. So under our task folder here, we have a separate folder with scripts. And this contains 2 scripts. So this demo subject.tcsh, this is a T-shell script that runs AFNI ProcPy for this particular subject. And the other file here is a run.sh. sh files are shell files that are used for queuing up jobs on the cluster. If you guys remember back to the first and second sessions, talked a lot about that, but that's basically just a script that says, hey, queue my subject.tcsh file so that it runs when resources are available, and so I don't have to watch it the whole time. So I'm going to go ahead and open up this demo subject.tcsh folder. So this is our this is basically a file to save some variables and then just run AFNI ProcPy. Everything in here could be run in a single line command, you know, in PuTTY here at the command prompt without any problem at all. But this has a couple of extra pieces of information that makes it easy to just alter a couple things and run a new subject. So the very top of this command here just says that this is a TCSH file. Pounds here are just comments. This just says, you know, when I last revised it, and then it sets up a bunch of variables that you're defining. So set means whatever string follows is a variable, and the equal says what that value is. So I have a variable called subject that is the name of my subject. And then I have top dir and results. This first one says, hey, this is where the subject data is located. I have a comment here, so if you're using different directories somewhere, you have to change just this line to make it work. That's if you're like copying the study data down. from our drive link. Then I set a new variable called results. So this $sign here says that whatever I write next is a variable, and when the program runs, it'll go through and replace that with whatever text is in that variable. So in this case, results is equal to this long file path. Here's our go no workshop, and then go no go subjects, and then it adds a slash, and then it looks and finds subject, which is up here. So it fills that in with demo subject, and then it adds the results folder, that last thing that we made in WinSCP. So that these two commands basically say, hey, here's where you can find the data, and then here's where you can put it, put the results. Okay, and then there's one more directory here, and this says this is where the stimuli are. So remember, this was all that, and then the results directory. This says, oh, hey, here's the onsets directory. Then there's 3 variables here that it sets that are used later. This first one says where the volume should be registered to. I'll cover that in a second, but in this case it says it to the value first. And then there's a variable here that says what the cutoff for motion we'll use later is in the scan. And then this says if how many, it's a variable for how many scans I want to delete at the beginning if I want to do that. And we can talk about that a little bit later. But these are basically things you might want to change, and so they're set as variables up here so you can edit them quickly. Then the next thing this command does is it uses cd, which is change directory, and it changes to the directory of the variable results up here. So it goes to all the way over to here, and here's results. And then it starts the AFNI procpy command. So everything after this point, except one last thing, is all AFNI procpy. So instead of setting all of these variables, every time there's a $sign, you could have just typed in your subject folder instead. But it saves us a lot of time this way.
So basically, when you have a new study and you're about to start this, it's really important that you get everything set up in like a directory structure, and you set it up exactly the same way in every person, and then you go into a template like this and say, okay, and change the little things. This is my top directory. It's called this. This is where the data are. And so you go in sort of with this template and make a few adjustments, and then you're not starting from scratch with this.
Yeah, the other thing that it really helps a lot with is, you're editing things in text. So if I'm like typing and then somebody comes in and I'm talking, and then my hand hit the keyboard, I've just destroyed my command, right? But if it's up here, it's really easy to see where you've messed up. And so that's another thing. If you're editing in the commands here, the chances that you might make a mistake down in this part that doesn't change from subject to subject is higher.
The other thing that you often have to do when, especially when you're first starting a study, you'll get everything set up and it won't go, it'll crash. So you kind of have to go into the guts of it to see where is it going, what is it, and make sure that everything is set exactly the right way.
Yeah. The first time there's a lot of errors. Now our demo set works perfectly. Right. I should make a harder set that just has some errors in it, but I took pity on everybody, so have a good example there. Okay, so it runs AFNI procpy, and then the last thing is it goes up a directory back to where it started. So let me go into a little bit of depth here. So the way it works is AFNI procpy. In Linux, spaces and tabs are treated the same, so it's not a big difference there. You'll notice all these lines end with a slash. That slash in this context just means treat it as though the next line is on the same line. So this has been broken apart. These hyphens are options for the program, and it's been broken out into different lines because, as you can imagine, if this was all glommed together, it's really hard to see what the options are really set to, and it's harder to read. So these slashes are just to make it easier to read. So there's a couple of things that it expects at the very beginning. This first option is subj ID, and that tells it what the ID number of the subject you're using is. And this is important because this is what it uses to name everything, name all the files afterwards, and some of the folders. And so it should correspond to your subject's ID for everything to be readable and easily recognizable. So that's all that command does. And then another really key important step in this is to tell it where it can find the data. So this dsets command tells it where the functional data is named for the subject. And basically each section of this has the subject directory in the AFNI subfolder, and then it's pointing to go no go 1 plus orig. And that's the data file that we moved in when we in the previous step. One thing to note about naming convention, plus orig means the file is in its native original space. Later we'll see other files that have things like plus TLRC, which denotes that it's been transformed into Talarak space, for example. But that's just a quick way that it notes what type of file it is. And so it gets 4 different files, and it knows to treat each input we give here as a separate run of our experiment. And just a rapid review, we tend in fMRI experiments to break things down into runs because it tends to give brief breaks for our subjects. If something goes wrong in any one of the runs, we have more leftover data, and it has a little bit better properties in the scanner. You don't want to run people for a very long time in the scanner if you don't need to. So it's easy to break things up into multiple runs. So by giving it 4 files, it knows that our experiment has 4 runs. And then this SCR overwrite just tells it that if there's a script already in the directory, it's okay to override it. This next line is tcat remove first TRs. This is a step if you want that correlates up to this variable here. If you need, for whatever reason, to remove a couple of TRs or like one pass through the brain in the very beginning, you can do that here. Typically in the past, neuroimaging, the first few volumes that we get tend to have the brightness contrast of the images is kind of a little messed up by just the properties of the scanner. More modern scanners tend to do 4 to 6 scans and then cut them out anyway. It varies by the scanner, but that's why that command's in there. Don't accidentally set this to something when you don't mean to, or everything will be off in your data file. Okay, so those are this section is kind of like setting things up, telling it where things are and how to approach the rest of the subject, where to find things, and if you're cutting things out ahead of time, that's where you do it. But then we hit this blocks command. Blocks command it's the command that helps orchestrate the order and organization of everything that follows. So blocks tells AFNI these are the things that I want to do, and this is the order that I want them to happen in. In neuroimaging, there's a lot of flexibility in which pre-processing steps you run when. There is not a strong consensus about which order to run things in, and it affects what happens to your data. So it's important for publication purposes that we are super clear on all of the pre-processing steps we did and what order it is if we want anybody to replicate our results. It's a broader problem in neuroimaging, but it's important to know these steps and how we do them.
And each of those are detailed in that page, right?
Yes, each of these steps are detailed in the page. I'm going to quickly summarize them because it's pretty long anyway. But you can get details and what all the different potential options mean later. So what I'm going to do now is I'm going to talk about each of these blocks and then special options we included. So T-shift here is the command that aligns the slices in time. So T-shift basically, let me jump to this first. So copy anat just copies the anatomy file to a place where it can find it. It'll need it later. So the way it works is when you're in the scanner and you have a scan, there's one image slice that the scanner takes, and then it moves through the brain until it gets to the bottom of the brain, and then it starts over. And you have to align all of those slices to one point in time to treat it the same. What's nice is more modern scanners, like the ones we use here, will take a lot of slices at the same time, so there's less of a gap in time. But T-shift says, okay, what do you want to treat all of these slices as? Like, what time did they happen? Because we have to put them all together. In this case, we've chosen the option. So options further down here can refer to these blocks. So T-shift align to says, oh, what are we going to align in time to? And what we set it to is to align everything in time to the first volume that we took. There's debate in the field about where you align to, but 0 is one of the more common ways you do it. Okay, so that's what T-shift does. Now, what align does is it aligns the anatomical image that we copied right here. So the anatomical image is a higher resolution image that identifies the brain structures, whereas the functional image is more about the flow of oxygenated and deoxygenated blood. So this is a high resolution image that shows us more of what the brain's anatomy is used. So we use that anatomical image to help move our individual subject's brain, which has its own particular shape and characteristics and size and thickness of the skull and all of these other characteristics. And we say, well, if we're going to analyze a bunch of subjects together, we have to have a common space for them to be in. So what Align does is it takes the anatomical image and it finds a method to best change the anatomical image to match the template we're using. In this case, we're using the Talairac template. And so what these later options with Align here set is what the way that the computer or the program automatically decides how to change the anatomical image to better fit with the template. So there's some options here that set giant move, which is somewhat humorously named, allows you to move the anatomical image a lot in the space to get it to align. This is pretty common because often the orientation of the image can be pretty far translated over. So this lets us, to try and get the images to sort of overlap, it lets us move it a lot. And then this hyphen cost says which cost function we're using, which is basically what iterative approach do we go through to find the way to squish the anatomical image around to best fit the template. And so these are all these methods are outlined in the help file here, but it's pretty extensive and very mathematical. You can get details about those different methods. This LPC function works fairly well. And then the last thing it does is it uses this add edge command, which basically gives a little extra space around the anatomical image when we're running it, and gives us some information about alignment later.
So the first thing that happens is the anatomical image, because you have to talk about it with imaging of lots of different subjects. The standard procedure is to put it into the standardized space, so you have an X, Y, Z coordinate. So what happens is the TLRC on the structural image is first put into it's warped and moved and squished so that it fits a standard X, Y, Z brain. And then this step is just taking the functional images and sort of mapping them onto the structural, right? So it's already gone through the TLRC, right?
I think that's actually the next step.
Oh, okay.
Yeah. No, it's okay. That does happen, but that goes a little bit later, okay. I think. So exactly what Eric was talking about there. So this TLRC step, this just kind of moves things around so that it's close to where it should go. The TLRC, the TALARAC step, what it does is that actually allows it to squish exactly to the template. So this option here, so ordinarily by default, it will it'll scale the image, the original anatomical image to better fit the template. It'll move it in spatially in space. It'll also rotate or pitch or yaw the image so that it rotates around. Those are called affine transformations. They change the characteristics of the image, but they don't change the relationship between each voxel in the image. This option, NL warp, allows us to use nonlinear warping techniques. Takes a really long time, but basically what it lets us do is it also lets us squish things that are in the brain that are bigger than the template down to the size, or pull and expand things out. So it basically lets us manipulate the space between voxels and the size of voxels a little bit. And what happens is, after the extensive amount of time it takes to find a solution to the nonlinear warping of the anatomy, it ends up with like a map, and dark things got smaller and light things get bigger. And so it's basically it's a map of how to deform that person's anatomy to fit the template. Then in the next step, they get merged together. This is VolReg, which is volume registration. So what this does is it says, hey, use this VolReg align to says, okay, use the first image in our series of functional images. So the functional images have an X, Y, and Z component and also a time component. So, each time we get a full volume of the brain, that's one TR. So this says, hey, when you're aligning everything, look at the first image we've got. This volreg align E to A says, hey, use the warp the epi images, the functional images, to the anatomical images, or sorry, align them. Then use the warp, the deformation map that we made here to squish the functional images the same way, with the idea being that our functional images are now in the same space as the Talairac template is, the thing that we're going to, the common space we're going to use to compare everybody. The reason we use the anatomical image instead of the epi or functional image is because the anatomical image has a lot more detail, so it tends to be more accurate. There's different approaches you can use. You don't technically need to use an anatomical image. This tends to give us the best overall results. Okay, so that's the volume registration step. So at this point, we have functional images that are in our Talairac space. Everything's sort of been aligned. Now, the thing about our neuroimaging data is, as Eric mentioned before, there's a lot of noise in the ratio between signal to noise. So the idea being that there's, in order to see the true neural activity, you have to deal with a lot of factors about noise. One way we deal with that is by looking at specific events that happened and having everything well synced in time. The other way we do that is we can apply a kernel, or basically just a method where we go through each voxel and we blur the signal of that voxel to the surrounding voxels. That might seem a little strange, but what it ends up doing is it ends up reducing sudden spikes in signal in one individual voxel, gets kind of spread over the surrounding voxels. The same with lower signal activation. So it smooths out random spikes that would make the imaging data confusing. The byproduct of that is it reduces the noise, but it also reduces the specificity of the signal, because when you see this amount of activation in this little cube in the brain, you know that some of that is from the surrounding information. So it's a little bit, we lose a little bit of our ability to say this is exactly where this activation occurred. However, it allows us to actually find our signal amidst the noise in the brain. So that blur step is very important. There's a mask command here. What this does is it gives you some options for if you want to cut out parts of the brain when you're running your analysis, you can do that. This setting just tells it to dilate the masks by about by one voxel essentially around the edges, and that's a thing that makes them a little more accurate. There's some elaboration of that. What it doesn't really do in this program is it makes these masks, but it doesn't really apply them. And what they found in various iterations of AFNI is that when you're comparing all of the subjects together, you end up taking out a lot of the information in the data that you don't want anyway, and masking can introduce some weird artifacts. So we generate the masks, so we have that option, but the only thing that we end up really masking in our data is things that are really large spikes way outside of the brain in our image.
That might be particularly good for somebody's got a tumor or a resection or something, which we deal with.
Right. You have a whole bunch of different options of ways you can mask in the program. This is actually the default that it does, and so we don't apply any of the masks in our analysis. And there's a bunch of explanation of that, but basically we don't really need the mask. But we make it anyway in case it becomes important later. Then there's the scale command. So different neuroimaging programs approach the fact that runs are separated in time in different files in different ways. So some programs will basically run an analysis for one run all on its own, and then do the next run, and then the next run, and the next run, and then combine them. AFNI, instead, what it does is it looks at each of these runs, and then to keep the signal level the same in each of the runs, for every voxel, it finds the overall mean level of signal in all the voxels in the brain. And then it says, okay, that mean level, I'm going to now make that 100. Then it takes distance in signal from that 100 point, and that ends up making each of these they have the same mean starting point. So then when they all have the same mean value, then they can be combined sensibly together. So it combines all of these after it's scaled. And the other thing that it does in that step is if there are any signal values above 200, which is huge, that's like many, many, many standard deviations effectively from the signal. It depends a little bit on your experiment. But basically, if it gets a value of above 200, it just sets it to 200. So it's Windsorizing the signal, technically speaking. But that helps reduce really random noise. We're not talking large values in change, so that's pretty reasonable. Okay. So then the next part here, so after it's scaled, now we're at this regress part. Regress stands for the regression, and the rest of this here, this is all of our regression commands. So the way it works when you're doing a functional analysis of neuroimaging data is you are essentially mapping changes in the oxygenation of the blood in areas of the brain. And when you've got more oxygen heading to an area, that means that area needs more oxygen because it's doing more stuff. This is an oversimplification. There's nuance here, but when we're getting our signal out in our neuroimaging data our fMRI data over time, we don't just see, oh, this event happened and then there's a big spike in oxygen. What actually happens is this moment starts, and then there's a gradual progression of oxygenation and deoxygenation that happens after that event. So to get all of that so that it's time synced up, we apply what's called the hemodynamic response function. But basically, it's a transformation that basically we've got this event that happens in time, looks at everything after, and then transforms things so that we're looking at changes in response to that event as they progress further throughout the scan session. And that takes some time to evolve. So one of the things that we have to tell it is with this regress basis multi, this command says the events that I'm about to talk about later, this is how to deal with them in relation to the hemodynamic response. So there's a detailed explanation of different things you can try. This block command is pretty standard. what that means, it's not necessarily block design, but basically what it means is when my event happens in time, go up to a value, in this case 1. So you have 0, and then the event happens 1, and then it goes for a certain length of time, in this case half a second. So that value goes up to 1, and then at the end it goes down to 0. And that's the structure of that event. This takes that information and convolves it with the hemodynamic response. So depending upon the length, you get this kind of protracted response in oxygenation that happens. And that's how we infer that this event that lasted this long, this change in oxygenation is due to, or is related to this event that happened in our experiment. I have 4 of these because these correspond to the 4 categories of events that I have in my design. One thing when we're running our regressions is that we want to make sure that if there's a lot of movement, that we don't include things that have so much movement that the voxel that we say we're talking about doesn't correspond to it. Motion's a big problem in running functional scans. People need to be as still as possible. Otherwise, when we say this voxel is related to this, but they've moved, 5 millimeters, this voxel here is now this voxel here. So we're very careful about that. What we do here is we censor any point in time in our analysis where there's a large amount of motion relative to the previous scan that we had. We end up cutting out that volume and the volume after it. And we regress out those so that they don't impact the analysis. We don't throw them out entirely, but they aren't included in these events. And that helps reduce the chances that something bad happens and we're not talking about the voxel we mean to be talking about. This is specified up here. Our maximum motion that we've set here is 1 millimeter. This second command is related to censoring as well. This, instead of motion, is censoring signal outliers. And it's a signal of outlier of 0.1, I think it's standard deviations, I'd have to check that. But the basic idea is that when you get a lot of motion, you tend to get outliers. You can also get weird changes in signal that aren't due to anything in a single voxel. This helps get rid of that, and it does it the same way by tossing out a volume. You'll recall earlier that we applied this blur to our data. These next 2 functions serve to estimate how much that blur affected our data with the size of voxels that we have and our kernel. It gets an estimate of how much the voxels have been blurred. There's 2 different techniques here, and this is really important because later, if we use techniques that generate cluster-based thresholds, and I'll talk about that more, but basically some of our analysis approaches that allow us to deal with the problem of multiple comparisons, which again I'll talk about more, these give us information that we need for that, because it basically discounts things based on how much we're blurring our data. Okay. So then we get to the actual meat of things. So this regress stim times tells us for each of these 4 events that happened up here, this is the file that you should use to get when that happened. You can either do this based on time in seconds, which is the stim times, or you can do it with TRs. But that's much harder to read and understand, and the ePrime file doesn't work in TRs. So it's easier to use this command. And this just reads in all of those events that happened in time for each of the 4 runs and says, okay, this is our regressor. So this one has 4 regressors. This next command goes through and says, this is what we're going to call each of those regressors from now on within this program. So I've labeled them CGO, CNG, IGO, and ING for our 4 different conditions. But this is what it uses in the next couple steps. This make ideal sum is a thing that's used in our quality control. But it basically summarizes, ideally, this is what the response in the imaging data would look like to the way you've specified your regressors. And this regress options gives us some additional information that also gets fed into our quality control. Okay, and last and most important thing. So this numGLT command, this is GLT stands for General Linear Tests. Num stands for number. So this command tells us the number of general linear tests we want to run is 7. So, and then GLT sim tells us what contrast we want to run for that test, and the label tells us what to call it when we're displaying it, what label it gets. So in functional neuroimaging design, you get activation that's related to events within your task. The problem is along with all of those, you get a bunch of other junk, like noise from your heartbeat and your breathing, and you get drift within the scanner's signal over time. Some of that gets accounted for in our regression anyway, but there's a bunch of stuff, and there's a bunch of unknown things that you're not sure where they come from, but aren't related to your task. So the primary way that you get around that problem is something called the subtraction method. And basically what you can do is you can take your image of interest, your event, and a summary of all the activation in those events, and then you can take another image that doesn't have your event in it, and you can subtract the signal in the two, but because that other thing has all the junk you're not interested in, you can compare them, and it basically removes all of that. It's another way of getting through noise. So one of the simplest ways to do that is to compare each of our events against a baseline. You can either make that a very particular event in your task, or you can make what's called an implicit baseline, which basically means you compare something against everything that's not an event in your design. And so you can do that, and then you can also do things that compare, let's say, one type of, like down here, our correct trials versus our incorrect trials, and that instead tells us relative differences in activation between two events of interest. But it also, through the subtraction method, removes most of that noise that's unrelated to our events. So the way this is coded is basically SYM just tells you you're about to start it, telling it what the contrast you want is, and then you use pluses to denote that this is our positive values are going to include here this multiplication sign times CGO, which is our label, the correct go trials. So basically what this says is positive things in this image I'm going to create are composed of this one contrast. And then because we don't add anything else, it's implied that that's tested against everything else that is not an event. So that's very easy in these simple contrasts. When we get down to the later contrasts, we have to balance things out by doing so you can incorporate, like here, multiple tests all together. The scaling on this isn't ideal, but the idea is that we can combine all of these. So correct go is positive, correct no-go is positive, incorrect go is positive, and incorrect no-go is positive. So basically anything that's in our task is activation, and that's all compared against our baseline.
Right, so basically what we're doing is for every voxel in the brain, we say if this voxel, the bold signal in this voxel is related to whatever event we're having them do. So in this case, they're push a button when they see a mouse, push a button. For every voxel in the brain, we say this is what the bold signal should look like, and it will play out over 30 seconds. And this is happening 50 times in this particular 50 go trials. So it's looking at what the expected response would be, and then this is the regression, what the probability is that what we actually saw related to the bold activity in this specific voxel. And then all this is other, whether it's involved in the go, no-go, whether it's differentially involved in the go versus the no-go. So that's the essence of what this regression is.
So this ends up coding for all of these different comparisons. So you'll notice when we're comparing two different conditions, and it's not just the implicit baseline, you code some being positive and some being negative, and that just sets up our comparison. This works just the same as regular linear regression, however you want to load things when you're doing contrasts. And then this last little bit is just housekeeping. If there's multi-threading, it lets you use multiple cores. And this execute command runs AFNI procpi. And then this command moves back up a directory to our base. So that's the overview of the AFNI procpi commands that we're sending. So next time I'll go through what the output of that looks like, both in terms of what the script that comes out looks like, but then also what the QC and results of this looks like as it's moving through steps that we're going through. So I'll try and show you the images of each of these steps so you can see what this command actually ended up doing. So I'll leave that for the next session. Are there any last minute questions about any of the stuff that I talked about? Okay, cool. So we can talk about more organizational things, but that's AFNI procpi.
Did you load AFNI on the Baker?
AFNI is currently loaded as a module on Baker, so all Baker hypercomputing clusters users can use module load AFNI to load it. And actually, a lot of these scripts load AFNI as part of them. Not in this case, but yeah, it's always a good idea to do that if you're starting to work with AFNI. And some of those details are in the earlier lectures as well. Okay, thanks.